Okay, welcome back, folks. In this video, we're going to start taking a look at nuclear division as well as the process of cell division or cytokinesis. So when I say cell division, I really do mean the splitting of one cell into two during the process of cytokinesis. It's what we've got to is the point where we're about to enter mitosis. And so the whole point of mitosis is to take the two copies of each chromosome that have been made during S phase, those are the sister chromatids, and to distribute them to opposite ends of the cells so that following M phase nuclear division or mitosis, we can engage in cytoplasmic division, cytokinesis, and divide one cell into two cells. So that's the process we have to look at. So mitosis is divided into 5 stages. Those stages are based on the changing appearance of the cell and the behavior of the chromosomes. So we have prophase, prometaphase, metaphase, anaphase, and telophase. Now notice that cytokinesis is not one of those 5. Cytokinesis is its own set of processes over here. So after DNA replication, cells exit S phase and they enter G2 phase, where the cell is making kind of its final preparations for entering mitosis. Towards the end of G2, the chromosomes begin to condense, and so we go from this very kind of ball of wool appearance of the chromosomes, I guess. They begin to condense into much more folded, compact structures, so the cell can actually separate them in the process of mitosis. The kind of transition between interphase and prophase is difficult to discern microscopically, and so it's not clearly delineated. Generally, we say that prophase has begun as soon as you can differentiate or you can distinguish individual chromosomes. Obviously, that depends on the resolution of the microscope. And so you may say you don't see any chromosomes, I may say I do, so it's a little bit subjective. But if we can see individual chromosomes, then prophase has begun. Because we've replicated the chromosomes in S phase, each prophase chromosome is composed of 2 chromosomes. They're tightly attached to one another. And so we have to look at the process by which we separate those. We're also going to look at the behavior of the centrosomes. So the centrosomes start out here close to the surface of the nucleus, and you can see the microtubules radiating from the centrosome, which remember is a microtubule organizing center. And so the chromosomes radiate away, the minus ends are embedded in the pericentriolar material, and the plus ends are radiating towards the periphery of the cell. As what's going to start happening is in prophase is these 2 centrosomes are going to start to separate. And as they separate, they continue to act as nucleation sites for microtubules that will become the mitotic spindle. You also get a dense cluster of microtubules called an aster that begins to form near each centrosome. So that's a little bit about the transition from interphase and the events of prophase. As we get into prometaphase, that's really marked, the start of that is marked by the fragmentation of the membranes of the nuclear envelopes. As soon as the nuclear envelope starts to disintegrate, then we're into prometaphase. At this point also, the centrosomes have completed their migration to what would have been the periphery of the chromosome, of the nucleus. And we can see we've got now fully condensed chromosomes. We've also got the microtubules that are forming the spindle. You can see the diagram down here, and you can see them radiating out into towards what will be the metaphase plate of the cell. These microtubules at the spindle are beginning to attach to the chromosomes in a region of the chromosome called the centromere. So let's take a look at centromeres and kinetochores. Top left here, you've got, you can see where microtubules are attaching to these green dots, and these green dots mark structures called kinetochores. We'll talk about those in a little bit. So the microtubules are here in red, and then the green dots represent kinetochores, which is assembled on the centromeric regions of DNA, and the centrosomes are here and here at opposite ends of the cell. The DNA is in blue. Don't worry about this diagram here. So the DNA in centromeres, which is where the kinetochores assemble, consist of simple tandemly repeated sequences of DNA called sen sequences. They vary considerably between species, so they're difficult to identify species to species. The sen sequences of all species that people have looked at is a special histone protein. If you remember the histones, there's a histone called H3, and that's called SEMP-A in humans. So SEMP-A, the sequence that recruits additional proteins to the centromere, and those proteins collectively form a structure called the kinetochore. And so it's to the kinetochore to which the microtubules attach. They don't attach to the centromeric DNA sequences. They attach to the kinetochores, which are formed at the centromeric sequences by the aggregation of this special histone called SEMP-A. And so SEMP-A is at the SEN sequences, and so SEN sequences are DNA. SEMP-A is a special histone H3, and then the kinetochore assembles at that site. So that process begins shortly after S phase. During prometaphase, the spindle microtubules are binding the kinetochores associated with each chromatid. And it's the forces exerted by those kinetochore microtubules, the ones attached to the kinetochore, which will gradually start to move the chromosomes into the middle of the cell so that we can enter metaphase. We take a quick look at the kinetochore structure down here. We see the centromeric DNA, and so let's just look at the big view here. So here's a centromere sequence. Here's a kinetochore on the left, and here's an attached kinetochore on the right. If we look at the unattached kinetochore, you can see that these fibrous proteins, there's a structure called a corona. If we look at the actual kinetochore over here on the right, which is attached to these microtubules, we see there's an inner kinetochore, and that binds the SEN sequences in the centromeric DNA. There's an outer kinetochore, and the outer kinetochore has these fibrous proteins which attach the microtubule to the kinetochore and therefore to the centromeric DNA microtubules. The centromeric DNA sequences. So as we look at a mitotic spindle, there are several microtubule populations. There are the kinetochore microtubules. Those are the ones which attach to the kinetochores, obviously, on the centromeres. There are then polar microtubules. Those are microtubules which radiate into the center of the cell, and they overlap with one another, and they interact with one another using a bunch of proteins which kind of associate them with one another. And then at the cell periphery, where we have the microtubule organizing centers, the centrosomes, are astral microtubules, and those are like arrays of microtubules, and they're shorter, and some of those astral microtubules interact with proteins at the plasma membrane, and we'll see why that's important when we look at anaphase. So during this stage, what we've got is the chromosomes kind of moving around due to the forces exerted on them by the kinetochore microtubules, and eventually all of the chromosomes will aggregate in the middle of the cell on a structure. Not really a structure, but it's a plane in the cell called the metaphase plate, and that's a plane which is our point in the middle of the cell, which is equidistant from each poles. The poles are where the centrosomes are. So we have the poles at each end of the cell, and then we have the mitotic spindle, and then in the middle we have the metaphase plate. And so at this point, the kind of microtubules attached to, think about it, these are two sister chromatids attached together, they're kind of being pulled in both directions by the forces. So they're kind of being tugged around in the middle of the cell, but they stay in the middle because the forces being exerted by each of the poles kind of balances out. And then something happens that triggers a process called anaphase, and we'll look at that when we look at regulating each of these stages. And so when we go to anaphase, which is the shortest stage of mitosis, the two sister chromatids separate. It happens very quickly. They separate abruptly and they move towards opposite poles of the cell. This happens in two stages. Okay, there's anaphase A, and that's where the chromosomes are pulled towards the spindles, towards the spindle poles, as the microtubules at the kinetochores get shorter. So at the plus end, we change the polymerization rate, so there's depolymerization, and that causes the microtubule to shorten, and basically the color of proteins which attaches the microtubule to the kinetochore basically migrates towards the minus end of the microtubule. There's anaphase A. Then anaphase B, so anaphase A there is the chromosomes moving due to the shortening of the microtubules. Then anaphase B, the poles actually move apart, so the centrosomes actually then start to move, and so then the spindle and the cell elongates, and so that's anaphase B down here, and so that's pole-to-pole separation. So that's probably due to the astral microtubules pulling towards the membrane, but also towards the polar microtubules in the middle of the cell pushing one another apart, and so there's a pushing force in the middle and a pulling force at the end. Now, depending on cell type, anaphase A can happen at the same time as anaphase B, and so these things are simultaneous, or you can have anaphase B following anaphase A. And then we enter telophase. And so telophase, the sister chromatids have arrived at the poles of the spindle, and so the chromosomes are pretty much right there at the centrosomes at opposite ends of the cell. The chromosomes begin to decondense, and so we go back to having interphase chromatin. The nucleoli start to reform, the nuclear envelope starts to reform, and then shortly after that we begin the formation of the cleavage furrow, and then we go into the processes of cytokinesis. So that's each of the stages in a little bit more depth. We'll come back and look at some of these again in a short while, so I'll see you soon. Bye for now.